Hi, everybody, dear colleagues, dear friends. It is my great pleasure to welcome you all into this special ICH activity, part of our ongoing mission to promote excellence in clinical hematology worldwide. And today's session is particularly inspiring as we are honored to host one of the most respected voices in our field. Our speaker needs no introduction to most of you, Dr. Vincent Rajkumar, professor of medicine at the Mayo Clinic. He is a global leader in multiple myeloma research and a distinguished physician scientist. His contributions have fundamentally shaped our understanding of plasma cell disorders, and his thought leadership has guided clinical practice across the globe. In today's activity entitled Insights into Clinical Trials and Research, Dr. Rajkumar will share his unique perspective on the evolution, design, and interpretation of clinical trials, and more importantly, on how we translate evidence into real-world practice to benefit our patients. We look forward to an enlightening session filled with knowledge, critical thinking, and forward-looking ideas. On behalf of the ICH, please join me in giving a very warm welcome to Dr. Rajkumar. Vincent, how are you?
I'm doing good. Thank you so much, Mohammed, for having me. It's a pleasure.
Excellent. So, well, you heard my introduction, and we would like, through this activity, to provide an in-depth exploration of clinical trials and research, highlighting their importance in advancing healthcare, but also would like to address the challenges and barriers faced by researchers, sponsors, and participants. So my first question to you, Vincent, is about, let's say, the foundations of clinical trials. Why clinical trials matter?
Thank you. That's a really broad question, and I think you have to step back and see where we are in the field. For example, if you take oncology or hematology, many of the diseases we treat do not have cures. Many of the diseases we treat don't have even optimal treatments. So when I entered the field, for me, the main thinking was that almost all of cancer therapy is experimental. We are constantly trying to see how we can improve outcomes for patients, how to do treatments more safely, more effectively. And clinical trials are the most important final component of that. Of course, early on, you have to have hypothesis, identify the potential treatments, the whole bedside to bench trajectory. But then once you identify a particular treatment that you think could help patients, then you need clinical trials to get to that stage. And if you just give a treatment to a patient outside of a clinical trial, what you think might be effective, you have no way of knowing whether it truly was effective or was a chance that the patient got better. You have no way of knowing, did I give the right dose? Could I have given a better dose? You have really no way of knowing what problems are caused, side effects by the drug as opposed to something else happened with the patient. So in order to truly know beyond your own guesswork whether something is working, you need clinical trials. And clinical trials need clinical trialists. So it's really good that we're having this conversation because right now there is a paucity of clinical trialists in the world in academic field because it is quite a challenging task to take a new treatment or even an existing treatment and improve it for patients. It's a long journey that starts with phase 1, phase 2, phase 3 trials, regulatory approval trials to change practice. Clinical trials are the foundation of what we do, and they have helped advance care. I mean, a good example will be multiple myeloma. I entered the field 1998, 99. We had 2 drugs that were approved from the 50s to the late 1990s. And then since then, we have like 20 plus drugs that have been approved for the field. What was a 2 to 3 year median survival is now 15 years or longer. And this is not by chance, it is by painstaking clinical trials, which requires patients, physicians, industry sponsors, regulatory authorities, foundations, all working together.
Wonderful. Thank you very much, Vincent. Uh, let's dig a little bit deeper now into the topic. And I would like to address with you on how to design a clinical trial from concept to protocol. And in another word, I would love to hear from you, and the audience would love to hear from you about the different steps in designing an ethical and hopefully effective and useful clinical trial. And obviously, what are the common pitfalls in protocol design and how to avoid them?
Yeah. So I think you have to say, like, so you have an idea for a clinical trial. How do you proceed? Um, Before that, broadly stepping back, there are two major types of clinical trials. One are trials that are designed and developed by industry, most of the time for regulatory reasons. But even within industry, there's a whole think tank of people who come up with the idea, who go through the same steps that we'll go through with an IIT. It's just that it's taking place in a parallel setup, and then it does come to leading investigators for their input before it finally becomes reality. And then there is the other group of trials, which are investigator initiated clinical trials. And they can be just single institution, or they can be multiple institution run by cooperative groups like the EMN or ECOG Akron or SWOG, where we come together to do these trials. I think the most important thing, Mohammed, is the idea. I think the first step is to be successful for the trial, for advancing care, is to come up with a good idea. Good idea requires a lot of background knowledge of what's happened in the field, where we need to go, and also a lot of time for people to think of a good idea. I can think of many, many trials which came up with really a fantastic idea, and that idea then moves on. For example, in the myeloma field, we at some point in time established that stem cell transplant was important. It improved quality of life. It improved duration without therapy. It improved progression-free survival. It improved overall survival. So we've been doing it for like ages. But then two or three investigators in France got the idea like, do we really need a stem cell transplant upfront for everyone? And that's the idea. And to execute that idea has been a 20-year journey of multiple trials testing the hypothesis. So the first thing is the idea. Once you have the idea, you have to make sure that you're able to put that idea pen to paper into a concept, whether you're asking a phase one question where you're trying to find the right dose, or a phase two question where you're just trying to figure out what is the efficacy and safety of this particular regimen in this dose, or a phase three trial where you're comparing your new experimental idea to the existing standard of care. In everything, you have to put together pen to paper and write down the concept. That's where a lot of thinking occurs in terms of who are the patients who are eligible, who do I want to recruit, what is going to be the sample size required to answer the question I have, what is going to be my primary endpoint, do I have enough patients and enough ability to actually have an endpoint of survival, or would I have to settle for progression-free survival or even response rate? And then you need statisticians to help you with assembling the right statistical plan. And a lot of input we get from patients and patient advocates to, is this trial something that would work? Is it something that's appealing to patients? Should we make it better in some way? Is it targeting the right population? We need to figure out what are the criteria we are going to use to adjudicate success or failure. And established criteria are there for most diseases. If not, you have to develop them as well. And then finally, you have to go through a number of reviews. It's your idea, you wrote the concept, you got the input from people, all of the participants and advocates, and including the drug companies that will provide the drug if necessary. And you've got your concept, but you still need reviews. So we have peer review of your idea after you put it to paper. And that peer review would be within your own disease group in your institution, could be a bigger group of experts who have to review the protocol. And then they're doing it scientifically and for the ethical reasons, an institutional review board. Uh, Finally, the trial is approved. You do need regulatory input. Sometimes you have to file for an investigational new drug application with regulators. And so it's a long journey. Sometimes it takes a year or two to go from an idea to the first patient enrolled. And once you get good at it, you can shorten that time period. Um, but it's a journey, and sometimes you've done a year of work, and then regulators or the reviewers don't like your idea, and you have to start from scratch again. So it requires a lot of perseverance, a lot of dedication. So the idea is the most important. And then on the other hand is the vision, where you want to go. Because that vision, if it's strong, you will be able to persevere and go to your end goal. And I usually say, like, you start with the idea, the midpoint is your first patient enrollment, and then your final success is actually when you have it published and as well as when the standard of care actually changes because of your idea. That's the long journey. So to really persevere in that long journey, you need that vision and dedication into why are we doing this? We are doing it to help patients. And if you have that, you will go through this journey. And you have a lot of colleagues who have done this before who can help you succeed in that regard.
Excellent. You mentioned several times already the ethical and regulatory considerations in clinical research. So here I would like to ask you on how to efficiently navigate the ethical landscape, but also how to navigate the regulatory hurdles across different regions, because obviously trials are meant to help patients everywhere, but whether we like it or not, regulations can be different from one country to another, local considerations, regional issues. So how to navigate all of these hurdles, because at the end of the day, we want to improve the outcome of the patients worldwide.
Yeah, great question, Mohammed. I mean, and it's a challenge, right? Like because of so many countries, access is something that's important to everyone, and you have to make sure that your trial helps more patients around the world. And I think we started off by saying the two big types of clinical trials based on, is it investigator initiated and done, or is it industry developed trials? Similarly, there's also phase 1, 2, 3 we talked about, where what is the question? Is it just dose finding phase 1? Is it to establish safety efficacy in one arm, there's phase 2, and then you have in simple terms, the control group to really find out head to head. That's the phase 3. But there's another big division of clinical trials too, which is, you know, um, what is your goal here? Is the goal to develop a new drug that's previously not available or a new regimen that's previously not available and get it to practice? Or is your goal, there's already many treatments and I want to figure out which of these approved regimens is actually better, or how to best use an approved regimen? And if I were to give you examples, we have multiple myeloma. I'll use that as a good example, 20 new drugs. We're still not curing this disease. So we endlessly need newer and newer drugs. Newer drugs in the same class to improve efficacy and safety over the existing previous drug, and new drugs in a different class that's never been seen or tested before. So That's one trajectory where you want to develop a drug, a new, brand new drug like a daratumumab, and you want to make daratumumab available to China, to Europe, to the US. So you have different ideas of how to make that happen. And then you have daratumumab is now approved. Should it be best used in the frontline setting, in the smoldering setting? Should be used as maintenance? Should we use daratumumab and not say bortezomib? Or, which triplet would you use or which quadruplet would you use? So those are questions after the drug's approved. So you have really two big questions, and the regulatory ethical challenges differ based on which question you're going for. So I'll do each one. When you're talking about a new drug, there are places, particularly if you look at between US and Europe, there are ways to get a really good new drug approved through what is called the accelerated approval pathway with just a single arm trial. And in some countries, that's okay, like in the US. In some other countries, it may not be okay. So we have to do things in parallel where we've already planned for, okay, I might be able to get accelerated approval with a single arm trial in the US. But for Europe, and to get the full approval, I still need a phase 3 trial. So you plan it like that, parallel tracks to make sure everyone gets access. When you do those trials, you have to make sure, am I comparing it to the right control arm? Am I using the right endpoint that will get approval? Because then regulators in the US might be willing to approve a drug with a progression-free survival endpoint. Maybe in Europe, they need overall survival endpoint. So how do I power my trial? Do I power it adequately for survival so that, do I make sure that the control arm that you pick is not only ethically okay, but also from a regulatory standpoint? Would regulators approve this control arm in the US? Will they approve it as a control arm in Europe? So a lot of considerations come up when you're doing these drugs that you're developing from bench to bedside across a phase 1, 2, 3 scenario, all the way to approval. When you're taking the other side, which is I already have all these regimens, all these drugs, how to best use them, when to use which drug, that's mainly sometimes it's done by industry, but a lot of times it's done by cooperative groups like EMN, ECOG, ACRIN, even in multi-centered trials, really to find out how do I best do this. And there again, you have the same considerations. You may design a trial that tells you how to use it, but you did it in the US. You didn't even have a regulatory goal. And there I always say, like, use your moral compass. If you designed the trial well, if you powered it correctly, if you had the right endpoint, and you had good control arms that you can be really sincerely pleased with, it will change practice not only in your country and your own institution, it will change practice elsewhere also, even if you design the trial for your own audience. So again, goes back to have a really good idea, brainstorm with colleagues to get it to a good concept, get input from patients, advocates. If you do your homework right, then the final product, even if you didn't have a regulatory goal in mind, will change practice elsewhere. And it's happened many times. The IFM trial was not designed for the US. It really looked at stem cell transplant early versus delayed. But my God, the impact it's had in the US in terms of how much we use frontline transplant is quite a bit. Same thing with the high-risk smoldering myeloma intervention. Even though Dr. Mateos' trial didn't change practice, it basically set the standard for Aquila trial and everything else later in terms of yes, we need to look at early intervention. It's not something we can sit back and relax because the patients are asymptomatic.
Thank you very much. So you have nicely described the complexity, the length of the process, all the challenges, and all of this takes a lot of time, a lot of resources, a lot of energy, dedication. So I would like to address here, because time is about money, procedures are about money. So I would like to address the financial and operational challenges in clinical research, especially in a globalized world, because running a trial in your institution is totally different from running a trial across 30 different countries. So my question to you is about what are the funding models for sustainable research these days, and how can sponsor optimize the use of resources, given also the increasing role of CROs and bureaucracy and procedures worldwide, with the standards becoming higher and higher, and requirements for monitoring, keep on checking this and this and this.
Yeah. So there are a lot of really important considerations here to take in mind. The funding models, um, when I started in the field, it was possible to have a trial funded by foundations and by even institutions, by the NIH. And that's getting increasingly challenging because the cost of a trial has gone up. Um, one of the first trials I did was the thalidomide trials in the early 2000s. Per patient, I was requesting only $3,000 for the whole setup. And so the whole cost of the trial was like for 50 patients, $150,000. And even that, we had to cut down to $75,000 total, which now, I mean, you can even 100,000 per patient, it's hard to find a trial. So the costs have become so high that most trials are funded by pharma, even though technically you can have single institution grant supported trials. You can have foundations like the International Myeloma Foundation or Leukemia Research Foundation or Leukemia Lymphoma Society, things like that that can fund a trial. You can have nonprofit groups that join together, do multi-institutional trials. Federal funding from government sponsors who want the best treatment for their patients. So the US, Europe, all of us have federal sources of funding, government funding that can do it. But that's all like only a small proportion of trials, mostly smaller trials. Really, the vast majority of funding is from pharma. And the pharma funding can come in two ways. One is they provide funding for an investigator-initiated trial, because they feel like you have a good idea. Yeah, that's a good idea. We didn't think of it, but it's a good idea, and perhaps you should do that trial. And we'll provide you the drug and the funding for that. That's a very, very common model, probably the most common model for IITs in terms of just sheer number. And the other one is where the pharma says this is so important, we need to hold the data. We need to run the trial ourselves, because if you make one mistake, you lose a paper. If we make one mistake, we lose a fortune, right? Like for them, it's money. So they will have their own CRO, their own internal trial. And of course, investigators will be involved, but the trial is run, the data is held, the I's are dotted, T's are crossed, everything by pharma. That's the pharma run trials, mainly for regulatory purposes. So, but the funding is still, for the vast majority of oncology trials, is coming from pharmaceutical companies. That should not go unsaid. And then a little bit comes from government resources, smaller still by individual philanthropists or individual foundations or individual institutions. Okay. Um, Now, a good way for a young investigator to really make their trial come to a reality. I will just tell you how I go about it when I get an idea. Is like, you're already thinking no great idea will actually get into become a clinical trial unless you have everybody on the table coming together. So I first make a list of the people whose buy-in I need. That depends on whether I'm planning a phase 1, 2, or 3 trials in terms of how many people I need to get in. But I really call, like, if I have a new idea and I think it's really important to execute, I run it by 2 or 3 colleagues to make sure it's not crazy and that this is a really good idea. And then I run it by the key pharmaceutical companies that I will need support from and say, is this a good idea? Will you remotely even support this trial? If they say like, no, we are really not interested, then there's no point in breaking your head. So you need their buy-in. And then who are the people who have to approve it? Like if it's a cooperative group trial, I have a side conversation with the steering committee people, with the CTEP, the NCI Cancer Therapy Evaluation Program, to see, is this idea something that you think you'll be interested in? Just to get an informal buy-in, and that really saves you a lot of heartache later, because you've not invested months and years of work only to find out in 2 minutes that this is not going to fly. So you want to make sure you get your initial read from everyone and people are supportive. Once I get that, then you write your concept and do the process that I talked to you about. Each step takes time. The idea and then getting the buy-in takes probably a month or two, and then you write your concept, and the concept has to be reviewed and approved by various people. But while you write your concept, you're still getting feedback from, you know, regulators, patient advocates, from pharma companies, and then you finish your concept, you get your approval, and then the full scale writing of the protocol. If you've gotten that far, then most of the time you'll be successful. So you're at least, putting a lot more work up front than having to withdraw it later. But there's no question, it's a long iterative, it just keeps going. It's a very long process. Many trials, despite your best intentions, take a year or two to go from, idea to first patient.
So, my last question, as part of this superb discussion, Vincent, you managed to get the right idea. You went through all of these challenges. You were successful actually in generating new positive data that hopefully will be impactful to patients. So my question here is, from trials to practice, how to bridge the gap? In another word, how do you do to communicate efficiently the results to stakeholders, to the public, the role of journal editors, policymakers, patient advocates, because at the end of the day, we want to make a difference in the life of patient.
Yeah, I will tell you, your work is easy if there is massive benefit. When there's massive benefit, you really don't need to persuade anyone. It just happens. It quickly takes over, and it's like, look at daratumumab. 2015 introduced, 2025, almost every myeloma patient's getting it, because it works, and it is also something that's safe and useful. Same thing with lenalidomide or bortezomib. These drugs work, and it's really not you take imatinib in CML or ibrutinib in CLL. You have many examples. Rituximab, drugs that work, regimens that work. Your sales, it doesn't need to be too much, because the benefit is massive. It's when benefits are important but require a lot of nuance in terms of patient selection, like who is the right patient who needs to get this, which is the right combination, which is the right dose for me to use it, what's the right dosing schedule. Um, it's a really important drug, but the benefit is only when I do it this way, in this setting. Um, you shouldn't do it this way, because if you did it that way, there's actually more toxicity. Like, you want to give CAR T, but you give CAR T to patients with a lot of tumor burden, you cause a lot of CRS and neurotoxicity, and so on. So there's a lot more than all these blockbusters, which don't require like major efforts to publicize. That happens. But this is really the bulk of our real improvements is not the blockbusters. That comes, but really, how do we get this message to everyone? I think all of us have an important role to play in that. Someone's worked really hard, got the evidence, run the trial, gets a good result, and That person is, of course, the first advocate for that trial. They present the data in papers and in oral presentations about how good it is. If we in the audience and those of us who have influence really think this is a good idea, then we have to help spread the word. And that can take the form of like CME meetings, seminars, like what you're doing right now. Like we are 8 o'clock on a Tuesday morning, we're sitting and recording because we want to spread the message. This is a shared responsibility of all of us because ultimately we want to help patients. It doesn't matter whether it's your research or not. It's someone's research, but you have to help spread the word for every good idea. So I take it on myself, like, whether the idea is mine or not is irrelevant. What is important is if this is important for patients, we are going to help disseminate it. Whether it's wearing the hat of somebody on social media, whether it's wearing the hat of an expert who's going and giving talks, or whether it's an editor who can solicit editorials, review articles, or even adjudicate the primary publication for making sure the right research gets disseminated. There's a lot of pressure on all of us to make sure that there's no conflicts of interest, that we are doing the right thing, that we are not doing our own biases. I like this, and therefore I'll just support this. This is my bias always. And so we have to really shed that bias and really be fair to the to everyone and go on. But I think it's a shared responsibility to promote it. And I partner with patient advocates, with foundations, with so many philanthropic foundations, so many nonprofit foundations that are patient oriented that can help us. We speak to patient family seminars to make sure patients have the information. And our meetings, like the International Myeloma Working Group or the International Myeloma Society, our national meetings are great forums to make sure that we spread the message to colleagues and help them spread the message then worldwide to patients.
This was quite incredible. Thank you so much, Vincent. On behalf of the International Academy for Clinical Hematology, I would like to extend our deepest gratitude to Dr. Vincent Rajkumar for his truly enlightening presentation today with clarity, depth, but also humility. He has shared with us some invaluable insights into the evolving landscape of clinical trials and research, reminding us not only of the science, but also of the responsibility we hold as clinicians and investigators. Dr. Rajkumar's vast experience, rigorous thinking, and dedication continue to inspire the global hematology community. We are very grateful for his generosity in sharing both knowledge and vision. Thank you as well to all of you for your participation and engagement. We encourage you to carry forward today's insights into your daily practice and also future research efforts. Until our next webinar, we wish you continued success and fulfillment in your professional journeys. This is the ICH around the globe and across the hematology field.